I want to extend a warm welcome to the 31st Annual Edlick Henderson Innovator of the Year Award Celebration. My name is Richard Chilla. I'm the Executive Director of the University of Virginia Licensing and Venture Group. Today we are honoring Dr. Timothy Showalter. I'd like to begin by welcoming Tim and his family, wife Shana, his daughter Lillian, his parents Ben and Jeannie Showalter, his in-laws Trudy and Eddie Lafrac, and his aunt and uncle Doug and Nancy Showalter. Unfortunately, Tim's other two children, Tessa and Brooke, had obligations and couldn't be here, but they're here in spirit. So, we also have other special guests today here who were prior winners of the UVA Innovator of the Year Award. I've seen some of them. I've seen Karen Ingersoll, Lee Ritterband, George Gillies, John Hasek, and Bill Petrie. If I'm missing others, I apologize. I was trying to do a little inventory around the room, but welcome and thanks for coming. And I'd also like to welcome Peter Grant, who's the chair of our LVG board. Peter, thanks so much for making the time to come here today. So, what are we celebrating today? The Edlick Henderson Innovator of the Year Award recognizes an individual or a team whose research discovery at UVA is making a major impact. As you'll soon learn, this is a most deserving honor for today's honored guest. Here at UVA LVG, our mission is to grow and guide university innovations while improving lives on grounds and around the world. Our tagline, if you've been to our offices, our tagline is where research gets real. It's on the wall of our offices on Preston Avenue. It's on our website and on the cover of our annual report, which you can grab complimentary copies of here later. But it's really much more than a tagline. It is our creed. Helping commercialize research is what we're all about here at LVG. I've been leading technology commercialization offices at major research universities for the past 16 years. And as I do this work, I continue to be humbled by the dedication, perseverance, and service to the common good here on grounds. We build upon the Great and Good 2030 strategic plan set forth by President Jim Ryan. It takes extraordinary effort for researchers who push beyond the traditional academic boundaries of teaching, scholarship, and research to engage with our office, to engage with LVG, to advance their research innovations past the laboratory or the clinic into commercial products that can benefit the public. As someone who's worked in commercializing new technology, both in industry and here in universities, this is really hard work. That's why I'm so impressed with the researchers at UVA who choose to do it. They are fulfilling the promise of federal taxpayer-funded basic research to advance the health, prosperity, and the quality of life of the nation. Now it's my pleasure to introduce Dr. Fred Epstein, my boss, Interim Vice President for Research. As UVA's Chief Research Officer, Fred is responsible for executing a research growth strategy, managing and protecting a $671 million UVA research enterprise. He oversees various university units that comprise research infrastructure, including our offices, the LVG offices, Office of Sponsored Programs, Research Development Office, offices for managing regulatory compliance and research safety, integrity and security, as well as animal welfare and environmental health and safety. Fred is the Mac Wade Professor of Biomedical Engineering and former Associate Dean for Research in the UVA Engineering School. He's been a world leader in advancing MRI myocardial strain imaging by developing and applying Cinedense MRI method. He's made important contributions to accelerated MRI methods and the methods and applications of preclinical MRI in mouse models of heart disease. In 2015, he was the only biomedical engineer invited to speak at the American College of Cardiology think tank. Formerly, Fred served as the elective chair of the American Institute for Biological and Medical Engineering Academic Council. And for his contributions to research and service, Fred is an elected fellow of three major societies: the International Society for Magnetic Resonance in Medicine, the American Institute for Biological and Medical Engineering, and the American Heart Association. So with that, Fred?
So it really is a tremendous honor to be here at this event. I myself am an inventor of technologies. Haven't yet reached the level of Innovator of the Year, so I'm humbled to be here and be speaking at this particular event. The Edric Henderson Innovator of the Year Award is the highest honor that can be bestowed upon a UVA innovator. Last year's winner, Mark Breton, helped revolutionize the treatment of type 1 diabetes. And the 2021 winner, Dr. Amy Mathers, developed a solution for testing wastewater during the COVID pandemic. In 2020, Dr. Rebecca Dillingham and Karen Ingersoll created a smartphone app that helps people who are suffering from HIV. I could go on and on. The list of innovators at UVA who have helped improve lives is a very long one. In fact, you can look at the board over here later on and look at all of the past innovators and what they've done, and actually Google some of them, and some of the contributions are absolutely remarkable of what people have done in the past. Really speaks to the contributions that have been made. One of the most remarkable things, which can sometimes go unnoticed outside the research community, is the important role that LVG, UVA LVG, plays in all the innovation. Rich's team at LVG works very tirelessly with researchers to discover an invention's smartest path to impact. And this is not a simple thing to do. So led by General Counsel Rob Decker, they navigate the legal and patenting process to protect the ideas that are developed at UVA. They connect and promote innovations to our partners and industry. Then they provide the resources to support the entrepreneurial and investment process. In the last 5 years alone, LVG has facilitated more than 1,100 invention disclosures, filed over 1,500 patents, signed close to 500 licensing deals, and helped create 30 UVA startup companies. Talk about impact and a lot of work and effort required to do all of that. Recently, and this is important, UVA LVG joined the NSF, National Science Foundation, I-Corps program. I-Corps is a public-private partnership designed to guide university graduate and faculty research scientists who have technologies and show them how to identify valuable product and service opportunities that can emerge from academic research, and to provide entrepreneurship training. I-Corps is really a remarkable resource for any researcher just getting into that idea of commercialization of their technology. If you're here and you're not quite at that point yet, Rich's team at LVG is always willing to answer questions to help you move along that pathway. So are you working in your lab on something you think may have commercialization potential? If so, give Rich a call. Give Josh Malden also a call. No matter what stage you're at, don't hesitate to contact them. We really are interested in moving technologies out of the research labs into the real world to make such an impact, and that's what the folks at LVG are here to do. Another one of LVG's important roles is managing the university's $10 million seed fund, which supports new ventures emerging from the UVA research portfolio. The investment team, which is led by Bob Creedon, who I believe is here, works closely with the licensing unit to identify opportunities. The team is governed by an oversight committee comprised of leaders in early stage investing and startup development who are all UVA alum. Investments from this fund are typically joined by other investors in each respective industry. And since the LVG seed fund supports UVA technologies, its portfolio spans several industries, including cybersecurity and medical devices. 2 years ago, UVA launched a second seed fund, investment fund, that was necessitated by a 50% growth in university research after the launch of the initial fund in 2016. And it has the same focus on companies founded to commercialize UVA IP, intellectual property, and operates under the same parameters as the initial LVG seed fund. So all of this is to say that we're very thankful for all the work that takes place at LVG, led by Rich. And we're also extremely thankful for researchers like Dr. Showalter, who are willing to step outside the academic realm and into the sometimes crazy and overwhelming world of entrepreneurship, all in an effort to improve people's lives and make the world a better place. And this is exactly what Dr. Showalter has done for women with cervical cancer. So Tim epitomizes what UVA strives for every day, consistent with what Jim Ryan always talks about, to be both great and good. And so congratulations, Tim. As Rich said a few moments ago, this is extremely well-deserved and really remarkably impactful for many people in the world. And with that, I'm going to turn things back over to Rich.
Thanks very much, and now I have the pleasure of introducing this year's Innovator of the Year, Dr. Timothy Showalter. Let me give a little bit of an intro here. So Tim was born and raised in Richmond, earned his bachelor's degree from Washington and Lee University before graduating from UVA School of Medicine in 2004. During most of medical school, he thought he would go into pediatric or medical oncology, but as a third-year student on an inpatient oncology rotation, Tim followed a patient who was receiving palliative radiation therapy, and he was subsequently introduced to UVA radiation oncologists Paul Reed and Jim Larner. It was then that Tim saw how he could use the technology to care for cancer patients. In his words, he was hooked. So that same week, he switched his career plans. He was placed into rotation in the radiation oncology department with Dr. Reed as his faculty advisor. Today, more than 20 years later, we are lucky to have both Dr. Lerner, the chair and professor of radiation oncology, and Dr. Reed, a professor of radiation oncology and otolaryngology. A little interesting side note, James Larner's father, Joseph, won this Innovation Award in 1993. So Tim just told me that today. That's a great story. So lastly, after completing his residency at Thomas Jefferson University Hospital in Philadelphia, Tim returned to UVA in 2012 as a professor and radiation oncologist. Tim focused on improving outcomes for cancer patients by evaluating advanced technologies for brachytherapy and external beam radiation therapy. And that's what led to his invention for women with cervical cancer that he's being honored for today. Tim's aha moment came at one of the last places you would expect, at a Lowe's here in Charlottesville. I think there's an interesting story behind that, and I don't want to ruin it. I'm going to let Tim talk about whatever he wants to say about that, but it's a pretty cool story. If he doesn't talk about it in his presentation, I'm sure you can get him to talk about it over a glass of wine downstairs. Last year's Tim's BrachyGel Vaginal Hydrogel packing system received U.S. Food and Drug Administration clearance as the first product of its class. It's expected to be in at least 50 hospitals by the end of the year, with international distribution commencing soon after. Tim recently sold his startup company, Advare, which was founded to develop this product, BrachyGel, to CQ Medical, a global leader in healthcare innovations. So the fact that Tim was able to do this is really remarkable, and it's not too often that an innovator can be the lead inventor, the co-founder of the company, leading the company to get FDA approval, and then leading the acquisition of the company. So Tim was able to be involved in all of that, and that's pretty remarkable. And most important is that Tim's invention is helping women with cervical cancer. This goes hand in hand with the mission we have here at LVG of improving lives. So, ladies and gentlemen, please join me in welcoming our 2023 Innovator of the Year, Dr. Timothy Showalter. Tim?
Well, thank you, Rich, for that warm introduction, and really grateful to be here and grateful for all of you for coming. I appreciate the recognition from the UVA Licensing and Ventures Group and from the broader university community. It's, of course, even more of a special honor to be here in the Rotunda, and I say that not only as a faculty member, but as an alum who's mostly been in here, getting a little bit of air conditioning on a hot day while walking around the grounds, so I am really grateful for that. As Rich already summarized, I feel fortunate in particular to have my family members here who, all came from the state of Virginia. I think my parents win the award for driving the farthest. So if my dad falls asleep at some point in the presentation, it's not because he's bored by the subject matter. I think it's because he woke up early and to go through all the tunnels and everything. And I'm also appreciative to have one of my 3 children here, though I think for today she's my favorite of the 3. But I will add that she did not have school today and is able to drive, the only distinction out of all of those 3. It's also really special to have such a diverse set of people from various aspects of life. There are not many opportunities where you can have colleagues and friends from the community and family members all in one place, celebrating something that you've done. And there are even fewer of those opportunities where it's for something good and not because there's been an unfortunate event that ended your life. So I'm really appreciative of that for this. I will also add that any innovation process like this, particularly one that is such a slog over many years, is really tiring over time, and so there are definitely ebbs and flow related to the energy for the project and commitment to the vision. And of course, there are people here, like many of my radiation oncology colleagues and biostatistical colleagues who directly contributed to the research project itself. But I would also like to thank those of you here who were willing to talk about things over a drink or on the squash court or in a morning workout. It's really I really appreciate that extra level of essential support to get through it. And with that, I'd like to transition to summarizing a little bit of what Advaray was or is and what we did, and give you a little bit of more detail about the overall journey. So first, for the Advaray team, I think maybe Michelle Wazorek may not yet be here, but when I refer to the Advaray team, I'm really talking about a tiny group of people. So there were 3 of us, all of whom essentially had full-time commitments in other regards that were at some time point in the origin of this company employed or actively involved on the company side. I met Michelle Wazorek around 2014 or 2015 when I called the previous Licensing and Ventures Group director and told him that I had no idea how to file a grant for the company. I had done that before, but I really had no idea what I was doing. And he gave me a business card or shared with me an email address for Michelle, who is a very experienced grants administrator and essentially had expressed an interest in helping innovators through these often challenging administrative aspects. And so she, from that point on, was instrumental in all of the headaches associated with grants administration and all that work, and helped me get through that process. Ultimately became the COO for the company. And then Amy Fansler, who unfortunately cannot be here today, who was a project manager and really came in at critical moments, like helping us work out the start of the clinical trial and who's going to print instructions for use kits for our devices that need to be shipped for the study. So it was really grateful to have them along the way. And then we were also fortunate that, again, through introductions via the Licensing and Ventures Group, we were ultimately connected with a group of really strong, nationally recognized consultants. I included the photograph of one of them in particular, Jordan Mickleby, who was connected to us via the contract design and manufacturing firm, Medical Murray, that we worked with. And at that point in the overall story of developing the product on the part of Advaray, that was the real moment where we got connected within an ecosystem that could help us through all of the regulatory hurdles and all the things that faced us. So I view that as a really critical step to really know who to ask. Just a summary of highlights along the way, and then I'll give a little bit more of a narrative arc to this and tell you a few of the pain points along the way. But just summary, of what happened at the very beginning to the end. This was an original concept that occurred to me based on a clinical need. I was essentially a busy physician, had never had any thoughts of any entrepreneurial programs or I would not have considered myself a candidate for an Innovator award. And I just had a frustration about what I had available for my patients. So that got me thinking about this potential project really as initially as a research activity. And then I was connected with a polymer chemist named Tim Long at Virginia Tech, and that came out of calling people and asking, who should I talk to on the chemistry side to make this idea happen. And that was initially a cross-institutional academic collaboration that was supported by the Ivy Biomedical Foundation here at UVA. And then subsequently, the company really took shape around 2017 when we received our first federal research award that allowed us to de-risk this technology, make some prototyping steps, and develop some preliminary data. And then we were ultimately able to get a phase 2 federal grant. Along the way, we got matching state dollars, and between those two NCI awards, we ended up having about $2 million in grant funding to work with. Sounds like a lot. It's really not a lot for developing the medical device, so we were pretty frugal along the way. The clinical trial was completed in 2022. The PI was Cara Romano, who's right here, and really proud that our department has such a strong clinical trials program and was able to complete this entire trial in that time. Highlights shown on the bottom, we're thinking about the company side. So in my basement one Saturday morning, Brachyfoam LLC was founded on LegalZoom. No idea, had never known anyone who started a company on their own before, so it was a pretty new process for me. One highlight pretty early on was that Michelle got to meet the governor. That was one of our state research awards, and that's when a moment where we realized that maybe we had an opportunity to move this further along. And then ultimately, this past year, Advaray was fortunate to have BrachyGel cleared through a de novo clearance pathway as a Class 2 device for the FDA, and then ultimately acquired by CQ Medical. What was really interesting about it is that the acquisition came down to the wire, and I was basically 16 hours away from hopping on an airplane to the ASTRO annual meeting, which is the main conference for radiation oncology. And I guess everyone scrambled and got their act together. What's shown on the right is the product actually displayed at the ASTRO annual meeting. And so it's really exciting to see after all of that stress right at the end that there's our product, and there were lots of clinicians asking questions about how they could get the product in their clinic. And this is what the device actually looks like. So just to orient everyone, I'll just tell you a little bit what this looks like in a kit. So it's basically packaged in a Tyvek pouch, and the product itself, BrachyGel, is a pair of syringes that looks clear like water, and it contains polyethylene glycol and baking soda and water, and it's some pretty simple chemistry. It comes supplied in preloaded syringes because my idea was that I wanted it to be really easy for physicians. I wanted it to be comfortable for patients. I wanted it to be easy for physicians to deploy. And the device includes a catheter that has some different channels and some engineering to make it work clinically, but essentially there's an empty bag, and the contents of the syringes are injected into the bag, and the gel solidifies within the vaginal cavity. As Rich said, this is the first product of its class, and so what that means if you've gotten a product through the FDA is that it's a de novo pathway, which makes the hairs on your neck stand up if you've done these before, because essentially the FDA is making history, right? So they're extra attentive to what sort of evidence that they need to establish safety and effectiveness. So I knew going into it that it was going to be challenging, and it definitely was, and I can share a little bit more detail with that later. Thankfully, it was ultimately cleared in, I believe, August of this past year, and then just a couple months later, we were acquired and CQ Medical will be both manufacturing and distributing this product, which I'm really excited about because the ultimate goal was just to get this out to help patients. CQ Medical is a relatively new company that resulted from the merger of QFix and Civco RT, which are two of the largest companies for positioning devices and consumable supplies and radiation oncology. So a surprise ending at the end of the year was I thought I'd put all that behind me, and I was fortunate to be on a holiday vacation with my in-laws and a whole bunch of family, and it had been maybe a month since I'd actively thought about BrachyGel, and maybe I'll share more context about the FDA clearance, but you can know why I was kind of like suppressing that memory. But I did get the surprise good news and an extra sweet ending at the end of the year for receiving this recognition for the Innovator of the Year award, and I've kind of been overwhelmed, but really I'm definitely grateful for all of the attention in my local 5 minutes of fame. I will say that when I made President Ryan's Instagram for this story, I was like blown away by how many text messages and emails I got from people that I hadn't talked to in years. It's really amazing the reach of that. So I don't know, it's pretty exciting time. I'm also looking forward to my solitude after all of this, but. So I'd like to step back and talk a little bit about why I felt that there was a clinical need for BrachyGel and describe a little bit about what the thought process was back in 2013, that this was something that I would want to devote so much time to. As Shayna has pointed out to me in the past, if you divide up my hourly for like what this worked out to, it definitely wasn't worth it, other than as a learning process and to help move the needle forward in terms of cervical cancer care. So first of all, for those of you who are not in the third row, middle there, like everyone else in this room, just to give you a little orientation for brachytherapy for cervical cancer, brachytherapy is the form of therapeutic radiation that involves a procedural element where you're physically placing a radioactive source into or right next to the tumor. And as you can imagine, when you're putting a source of radiation right next to the tumor, you can get a lot of radiation dose to that tumor, and you can also do it in a very safe fashion because you've avoided aiming a radiation beam through the tissue and you're delivering it right to the point. Radiation, if you're not familiar, falls off according to the inverse square law. And what that means is that even like millimeters or centimeters away, the amount of tissue exposure to radiation is really minimal for this form of treatment. It is the one part of our discipline in radiation oncology, so we're all boarded to deliver therapeutic radiation, but it is the one part that requires us to perform a procedure. It's definitely not a complex surgical procedure, but it does involve some equipment. It can be frustrating, and it does require direct one-on-one time with the physician and that patient. It's a really important treatment for cervical cancer. We do have a problem with underutilization in our country for this, to the significant survival detriment to our patients. And part of that is because it is so resource intensive. It's just not available broadly. About 10% of radiation oncologists perform brachytherapy as part of their practice. The overall course for patients involves coming to a treatment center. Often they're driving hours because, say, they're coming to UVA from far Southwest Virginia or even Tennessee, and they're coming here to get this sort of treatment because they can't get it elsewhere. I'm very proud of the program we have at UVA. We actually have a CT scanner that literally slides across the floor in order to deliver high technology radiation for these patients and to do it in a fast format, so that the idea being that get people back to their life. It's a very compelling patient population. These are often women under the age of 50. The typical, like a couple of the patients that I've talked to in the past about like the convenience struggle with this and like how burdensome the treatment is. Let's just tell you like a couple typical profiles. So one, like 28-year-old like chain restaurant waitress from Lexington, Virginia, 2 kids, single mother, has to come to UVA for treatment. The brachytherapy procedure occurs at the end of a course of external beam radiation and chemotherapy. And so people are pretty tired, and they've put up with a lot of side effects. And so when we get them for a 2 to 3 week experience where we're doing 4 to 5 outpatient procedures, they're at like the low point right at the end of treatment. And it can be a really painful and uncomfortable and awkward position to be in as a patient. And so to me, it's always been a really compelling patient population to serve. As someone who came to UVA in 2012, I will tell you, I felt pretty confident about my ability to perform this procedure. I had been in Philadelphia. I was in practice for 3 years. I was pretty cocky. And I came down here, and I had maybe done 25, 30 of these procedures per year. I had taken care of like a wide variety of patients at where I was before, which is Thomas Jefferson University Hospital. And when I came here, the guy before me left like 2 days before I showed up. And I was like the only person doing brachytherapy procedures. And I think that year I did about 150 of these cervical cancer procedures. And I had a new respect for how much work it was and how challenging it was to get people through this treatment course. I also had a new appreciation for how frustrating some of the limitations of our procedure was. So let me frame a little bit about like the implant procedure and the packing portion. So let me start with the end of the procedure. Okay, so I'm going to explain to you like what's pretty typical for a radiation oncologist. So if you look in the middle here, contour structures. So when I'm contouring structures, I'm sitting in front of a nice large computer monitor. I might have a higher grounds coffee cup next to me, and I am carefully going through and telling the computer what is tumor, what is normal tissue. And then I'm working together with a physicist to do very elegant computerized treatment planning. We can use algorithms to optimize the therapy. We even have artificial intelligence-based contouring, so it's very high technology. We can fuse multiple imaging modalities on top of each other, so we're working with really large amounts of data. And then ultimately, at the end of the case, we even have a robot that literally pushes a radioactive source down each of the channels for the brachytherapy applicator, and so it all seems very high-tech. But the parts before that are decidedly low-tech and can be very frustrating. So if you look to the left, the place applicators and insert packing materials, we have a limited set of sizes for our applicators, and they're in a uniform geometry. Cervical cancers don't come in a uniform geometry in terms of tumors. They have all different sorts of shapes, and everyone's anatomy is different. And so we often feel like we joke that we feel like MacGyver, in the brachytherapy suite, but it's really true. And it can be frustrating, and you've got a patient who's wide awake experiencing this the whole time. And it always struck me that at the end of placing the brachytherapy applicators, and just before we get our CT scan and start doing the computerized treatment plan, that we place this vaginal packing. And it's an important step to protect the bladder and rectum. And at the time, I was really disappointed by the options that were available to me. And option one was basically taking a strip of surgical gauze, and which is widely available at all hospitals, wrapping it around forceps, and then placing it in an awake patient through multiple insertion movements, and that's really uncomfortable. It also is recognized to be relatively poorly performed occasionally by physicians. So you can be a well-trained brachytherapy expert, and some days you're just going to place the packing incorrectly, and it can be a substantial time delay and cause of discomfort for patients to go back and redo it. There also was a single commercially available product called the Elatus balloon that I wasn't happy with the performance of, and it also came at a very high retail price, and there was no published data. So if you're asking a healthcare administrator to approve a purchase for you for a new device, you really need to have evidence to show that it improves comfort for patients or has some sort of a positive outcome, and I didn't have that. Thankfully, UVA I think was sophisticated enough to interpret the arguments in favor of the balloon, and I was able to get it passed. But it always struck me, and we have used that at UVA for years, but it always struck me as really an unnecessarily burdensome process, and I really wanted something that I thought performed better and better matched the level of technology. I will also add, it's not just me that finds packing frustrating. I found a quote years ago from one of our colleagues, Mitch Kamrava, who practices in California. California, from a reviewer article that says vaginal packing is the most technique-dependent portion of the procedure, and it tends to be the most uncomfortable part for the patient. So it just frames it for you that I just described for you contouring tumor volumes and normal anatomy on MRI and CT scan, and we're saying that this is the most technique-dependent portion of the case. So I felt that there was really clearly a problem. So one day, early Saturday morning, we had a simple home repair that needed to be done. There was a little gap in the masonry and insulation in our basement for an external faucet. So I probably had some of the kids in a shopping cart or something, was in Lowe's and was walking through the aisles, and I ended up needing to buy a can of spray foam. And I used it for my home repair, and then I sprayed some in a trash bag afterwards, and the thing kept expanding. And I just thought to myself, I use this for this little job, and then there's this thing can fill any size container you have. And so I had this idea, there should be some version of this for vaginal packing for cervical cancer. So ever the young, eager guy without any gray hair in 2013, I hammered off an email to David Chen, who is in biomedical engineering and runs the culture program, and said, he sent this to me recently. I don't think he was trolling me. I think he was celebrating this with me. But he said, I think the design, this is what I said to him, I think the design is readily commercializable, but I just need a collaborator to refine the packing idea and to develop prototypes. So I figured I'd have this wrapped up, within the next couple of years or so, and move on to the next idea. And that was in 2013. So. And just to frame things, so this was my life in 2013. So to show you how long this idea has been going on, the one in the middle for this adorable 3 right here now has all of her teeth and could have driven herself here. I don't know how you got here today, but so it was a pretty wild time, and I'm of course grateful to Shayna and to the kids for not giving me too hard of a time and supporting me during this long process. So a little bit more details about what it entailed to get this, I'd say even starting the race and then ultimately across the finish line. So ultimately I was connected with a wonderful polymer chemist, Tim Long at Virginia Tech. He's now at Arizona State University. And I could speak radiation oncologist, but I didn't know how to speak chemist for sure. And I also didn't know how to speak biomedical engineering or regulatory or any of the other bits of language that one would need. And thankfully, Tim has been down this path many times before and was able to help me translate what I was asking for in terms of performance characteristics of the gel into some measurable milestones that we could start to work off and develop some preliminary data for. I was looking for a gel basically that could absorb radiation because we needed to use it for as an adjunct to brachytherapy treatment. I needed to be able to function sort of like gauze or other devices so that it could actually move away the bladder and rectum. And I needed it to basically, be biocompatible, not cause any irritation, to look good on imaging. How you actually translate those characteristics into something that an engineer or polymer chemist can act upon was this dance of trying. I felt like we were gesticulating and talking slowly to each other, but finally we got there. The one story that I will tell you that was the most inventive, that I'm still amazed that it worked out this way, is that our decision, the way to pick modulus, which I didn't know the term for before, but essentially is how much force can that material displace, is sort of the basic version for it. So we had to pick a modulus for how much pressure we wanted for the gel to exert. And the reason why we needed to do that is that I needed it clinically to be able to form in position and push some soft tissues away, even just a few millimeters. And the way that we found this is we found an article from the 1970s from physiology literature about the pressure generated by Valsalva, which is basically if someone tells you to if you're squatting weights, or if someone tells you to bear down, that's what Valsalva is. And so someone had measured what the pressure on vaginal mucosa is somehow in a cadaver study or something. And that's the shred of evidence we used to pick this. I'm just telling you, we were grasping at straws. But it turns out when we started working with cadavers and ultimately human patients, that's a pretty good guess, and I'm glad because we had no other information to go on. So I just want to mention a little bit about what Tim Long had to put up with me when I met him. So as I mentioned, I shared my email to David Chin, that, I was sure that I had this idea for polyurethane foams, basically. And so I was finally connected with Tim Long and told him I have this idea. I need you to make me a polyurethane foam that I can use for this clinical application. He politely informed me that polyurethanes are known carcinogens, and that foams won't work because what makes it a foam is that there's air there, so that's going to compromise the radiation absorption, and that I need a hydrogel for sure. So 2 things occurred to me. One is I'd already filed on LegalZoom for BrachyFoam LLC. And I don't know, I'm not a brand expert, but I'm pretty sure that BrachyFoam, it's actually not a foam, it's a gel, or never mind, it's not a good slogan, you don't want that on your labels. So that was an issue. And then I quickly learned that it would probably be better to not tell Tim that I was actually a chemistry major in college, because there was no evidence of it in our conversations, and it was just safer for that. I think that the first big step for us is that we were able to really build our collaboration around the Ivy Biomedical Innovation Fund Award, and it really enabled us to build a cross-institution collaboration. These are pretty rare to have a very committed collaboration between Virginia Tech and UVA. We were able to have a postdoc that was split between our institutions, named Nick Moon, who really drove a lot of this initial preliminary experiments. There was a lot of work that went into figuring out exactly what hydrogel recipe we should use. So all the experiments of looking at different concentrations of sodium bicarbonate and how much water should be in there, and all these steps along the way. We had a medical physics colleague, Bruce Libby, who was here, who was very involved in helping push that along in terms of making sure that we could run experiments in a timely fashion, and that we had our imaging done. And then we got important contributions from 2 of David Chin's students, Justin Traynor and Wallace Giles, who have left the university and are on to other things. Even Michelle's daughter helped us print 3D phantoms that Justin Wallace designed for us. And so these were really critical steps for a physician who wanted to push these forward, to have the technical support to fill in the chemistry details and to fill in the engineering work that we needed to do to even run our experiments and show a proof of principle. What we ultimately landed on was the Michael-Thiol click reaction. And I had never heard of this before, I will tell you. I was aware of polyethylene glycol, so that's pretty common. If you've ever heard of someone taking Miralax before, for example, that's the ingredient in that, so it's pretty widely used, pretty comfortable that it's non-toxic from the beginning. And then there's baking soda, water, and then there's one chemical that no one had heard of before, which is called Thiocure. It's provided by a single manufacturer in Germany, and thankfully we were able to confirm that they'd already done a lot of the biocompatibility testing. So even though we had to repeat all that, we were confident that this was a chemical strategy that we could work with. We published one paper that essentially went through how we defined our recipe, and we were able to establish a few important performance characteristics. One is that we knew if we put enough baking soda in there, it would form rapidly. So that was one part. We knew that we could generate enough modulus for this, which I had been taught about from Tim Long. We knew that there was no inflammatory reaction, basically through a preclinical assay that we did to establish. So we knew there was not an irritation caused by this. And then we were able to perform imaging that showed that on a CT scan, you could distinguish the gel itself from a brachytherapy applicator, which is the device that actually holds the radiation source, and from water. So we were go time and ready to go. We then moved on to comparative cadaver experiments. We wanted to get a sense of how this would perform. And this was done in the UVA cadaver lab. It's a soft-cured cadaver lab. I don't ever want to spend time in there again. But I will say it was an amazing resource for us, and the embalming process specifically creates cadavers that can be used for surgical simulations and training like this. And because the tissue interacts more like live tissue, it's a great resource at UVA. And I'll also add that we benefited immensely from coordination from the director of the cadaver lab and even from Caitlin Connolly, who's back there, for transporting these cadavers. You would not believe how complicated it is to transport a cadaver from the lab to a research imaging facility. There are like mortician services involved. It's a whole thing, but it's really amazing the like extra mile that people went to make this happen. We learned a couple things around that time. One is that we were able to establish with the 3D printed phantoms and the hydrogel itself, that final recipe that we had come up with, that the core idea of this, which is that a hydrogel can expand, this sort of approach can expand to fill any shaped cavity, worked. And shown on the right here is a comparison in this 3D printed phantom over here, which was generated by UVA BME students, that we could show that actually the gel would go into those spaces. And if you compared it to balloons, you notice that the balloon does not go into that spot right there. And that's a good, we felt that was a good test system for what it would look like around a cervical tumor in the upper vaginal area. The key finding number 2 is that based on the cadaver studies, based on a variety of things like the reaction has to happen in an enclosed space. So we figured out really quickly in the like awkward situation with cadavers that it was going to be a complete non-starter to inject gel into anywhere near a patient because it doesn't react immediately and it's just a mess essentially. So we quickly spun up the idea for prototypes. So at the time, now a radiation oncology resident, but at the time a research assistant, Michael Shad, helped do a former UVA BME student actually, helped come up with some basic prototyping that we had developed by the end of the STTR. So we essentially had envisioned like an epoxy gun type setup that would go into a plastic bag. And then we worked with Medical Murray, who we'd contracted at that point to develop some basic prototypes. And those are shown here, like in some of our earliest experiments of that the gel could form in the bag and could take the shape of a container. We then were fortunate to get a phase 2 research grant. And basically this was a large enough award that we were able to move farther along in designing the actual delivery system that became our product. So far along that we were able to enter a design freeze and go through design verification and validation testing. And that was all done with Medical Murray. And we were able to test those in phantoms. And then we verified performance in cadavers, again with the final version, just so that we were confident that all the kinks were worked out. And then we also conducted biocompatibility testing and chemical characterization, which were 2 things that the FDA added for us since we were that more challenging de novo pathway that I mentioned. And then ultimately, the most important part is that we validated performance in a clinical trial. This was conducted entirely at UVA, huge team effort from the entire department and pretty remarkable progress. It was not considered required for our FDA clearance, but I viewed it as an important step to generating evidence to confirm that this improved patient care and is something that I would want to recommend for use in clinic. We designed it to be streamlined, so basically from the time you open the kit, we want it to be obvious what should be done here. We want it to be designed in that sort of a way so that it makes sense for a physician based on other things that they've used. It's got a Y connector in it that connects to each of the 2 preloaded syringes. And then ultimately the contents of that are injected into the therapy bag there. At the end of the procedure, it's simply removed. There's a saline flush port in case there's a need to break up the gel a little bit at the end of the procedure to remove it. But I'm really proud that it's a pretty seamless process. What we sacrificed in this design is that it doesn't have a long shelf life. So there's actually it has to be stored because it's an aqueous solution in refrigeration. And right now we only have a 6-month shelf life, and we anticipate being able to extend it to 12 months in the next year. I was pleased with how things looked in general on imaging. What's shown is really sort of all the steps of our research. We've got how things look on like a pelvic procedural training phantom. It's like a little mannequin essentially, and on the middle is MR images from the cadaver from our NIH grant. That just shows that it looks it's distinguishable readily to a clinician on an MRI. So when you're doing treatment planning, it's really important to have that. And then shown on the right, we had a whole bunch of images from all those clinical trial subjects, each of whom had 5 treatments. So there was a lot of data from that. I think the biggest part of this overall research program, and I'd say when I think back to this, like I think what I'm most proud of, and when I look at the evidence packet that was submitted to the FDA, is this clinical trial. So I've shown here some of the main people that played a really critical role for this trial. I will also add this was definitely a team sport. So there were many more people who are not on here. I'd like to highlight Cara Romano, who was the PI for the trial and who presented the results from this at the American Brachytherapy Society annual meeting last year. And Gina Petroni, who's right back there, I think wearing the same jacket actually. So I'm sorry, I should have checked this first. Who was our biostatistician. And for those of you who are not in the clinical trial space, just to know, Gina is a world-renowned expert in clinical trial design, and this particular design with where patients are randomized to the order of therapy of packing, so it's standard packing versus BrachyGel packing, was a really critical way to conceptualize this, and I think that was probably Gina's idea actually, but she has a very good job of listening to what the clinical question is and asking a lot of questions, and disagreeing when she wants to disagree too, but ultimately coming up with a great solid design like this. And it feels really good as an investigator to have a clinical trial with her, because you know that it's going to be well designed and very rigorous. And then I've shown here Song Wood on the bottom right was worked tirelessly as a clinical research coordinator for this, and even heroically at the very end for a lot of the data capture that the FDA came back and asked us for. And then Bruce Libby, who's now at Moffitt, was really essential in the early stages, and the rest of our medical physics staff really stepped up and helped out with a lot of the clinical trial as well. The primary endpoint for this study was radiation doses essentially to these organs, because that's what the job of this material is to protect those organs. And there were a total of 20 subjects enrolled. What I'm perhaps most proud of is that due to a conflict of interest management plan, my involvement in the trial needed to be at a minimum. It's just sort of basic good practice for research. And my colleagues really stepped up in a major way and performed these procedures. There was a really high rate of data completion for the surveys that had to be filled out by patients, physicians, and physicists. And I will also add that the one major protocol violation was me, because I forgot the patient was on study when I covered a case, and forgot to use BrachyGel. And in a weird way, I think during the FDA discussions, it added a level of credibility that since I was representing the company, that I was the one who made the mistake and didn't use BrachyGel for some reason. And then I'd say that one of the best moments that happened for me, and I had this happen on more than one occasion, is that the patient, it's not blinded, they know what's being delivered that day. And the patient, as I started to perform the procedure, remarked to me just voluntarily, "Oh, good, I'm glad it's a BrachyGel day." And as you can imagine, having spent so much time on this, it's great to know that all that work is benefiting some people. And I was in this situation where I didn't really know any of these patients very well, because they're Cara's patients primarily, and so it just felt really genuine and made a lot of this worthwhile. Primary results for the trial, it's a really small study, but I think it obviously was strong enough evidence to support the FDA's recommendation. And they looked at this data every which way and every different sort of spreadsheet format, which was, and I appreciate the UVA team's contributions for this. But the gist is that there was no significant difference, and essentially the conclusion is that based on the evidence that we have, it looks like BrachyGel and gauze performed similarly in terms of protecting the rectum and bladder from radiation doses, and also probably almost as important, making sure that we give a similar dose to the tumor. It's actually sort of proud that the average radiation dose to the tumors for BrachyGel treatments were higher than for gauze treatments. There were no major toxicity differences between the two groups. There was a trend towards improved patient comfort, or rather, as better restated, less severe discomfort with BrachyGel over gauze, and the physician ease of use generally trended towards better with BrachyGel, which I think when you're rolling out a new product that people are just getting familiar with, I think says a lot about the design, and some of that was luck and some of that was hard work and finding the right people to work with. So at the end of that, thinking about what we did to develop the product, this is a slide that I had in the pitch deck. We never ended up raising money. I don't know if you don't like the slide format, maybe that's part of the reason why. But essentially, this is the core idea for like why I think there was a case to spend all this time developing BrachyGel, is that I wanted to improve patient comfort. So that clinical trial result showed that. I wanted to have a more customized fit. So the idea was that around the hot spots around the tumor, I wanted to be sure that we had a solution that could get into all of the small areas and protect the vaginal mucosa, protect the bladder and rectum. And we showed that. We showed that in bench testing, and I think the dosimetry results bear out. I wanted to develop something that I felt could support outpatient procedures and just make it less uncomfortable for patients who are experiencing something with essentially oral sedation, but also less frustrating for physicians, because as I mentioned, there's this real problem of people not wanting to perform brachytherapy procedures. And then at the end of the day, I wanted to be able to tell a value story. So I wanted to be able to say physicians who want to use this technology to be able to look at the results and have confidence that we're actually improving a clinical outcome, which I think what this is all about. So I think we were able to generate that evidence, and that research manuscript will be published later this year. Now I want to talk about the FDA application process. So I actually don't know the person in this photo. I found this on a Google search. I did sad, alone guy. And I just couldn't think of a better theme for how I would summarize the FDA application process. So just to frame it, this was all unfolded. We filed in the end of August 2022, and then we ultimately at some point in 2023 entered the interactive review process, which is where you wait for a long time, and then you get your questions, which I will say all of which were very reasonable questions from the FDA. You can tell they understood it. And they give you 36 hours to turn around 100 man hours worth of work. I mean, it's that's the classic sort of thing. So I had this body posture in the Reagan airport one time, sitting on a nice restaurant, trying filling this out instead of talking and enjoying the evening, those sorts of moments. So I wanted to just summarize the process rather than getting to the nitty-gritty too much about the de novo process, and I just wanted to provide some numbers here for context. So one is that it took exactly a year. So they have a deadline August 22nd, 2022, and you get your decision letter by August 22nd the following year. $40,000 in filing fees. That's not that bad. We had a small business designation for this. During the interactive review period, we had at least 10 unplanned bench testing studies, which we hadn't budgeted for and did not have money for and had not planned to do, and we were just pulling favors to expedite things with consultants and various folks. Our final application had more than 100 PDFs. I would say there were hundreds of unplanned personnel hours during this time period, not all of which was mine or Michelle's. Some of it was on the UVA side as well. In fact, there's one weekend that I know that a lot of folks spent a lot of hours getting things done. I showed you the picture before. I did not have gray hair before this started, and I've got a lot now. I think it's thousands actually. And then we did run a little bit over our grant funding by this, and I think for folks have had multiple rounds doing this, people would say, running over $2 million is no big deal, but we were just so close. So fortunately, we were able to cobble together essentially a loan to get that through. And then I will just call out there was at least one night lost during a weekend getaway at a fancy hotel where I was sitting there catching up on spreadsheets. So again, thanks for the support for getting through that. And then one of the nicest things was to receive this letter. The FDA has a low-key way of giving you good news. So basically, they grant the de novo designation, but it's not like a college acceptance letter where it's congratulations or anything. So I had to read this a few times, but this is one of the best letters I've received in a long time, that essentially we had received a class 2 de novo designation. It's a pretty long letter. And the reason is that they're basically providing descriptions and details about not only our product, but what they would look for future products in this same category. So then I'd like to talk about the acquisition. So shown here on the left is Michelle and I in Chicago for the Astro annual meeting in 2021. And the reason why we were there is that for a time we were pitching to investors essentially and thinking about raising a round of funding and doing some of the commercialization ourselves. And at one point we pitched to Civco RT via Zoom, and we were approaching them as like a strategic partner essentially. And what they explained to us is they liked our pitch, but they don't do strategic investments, they do acquisitions, and they would like to meet with us like a month later. So we hopped on an airplane like titans of industry, like ready to go, close the deal. And this picture was snapped. We thought to snap it like right after leaving the conference room that they had reserved for this meeting. Just confident that this deal was about to close. You can tell the time because I'm holding a mask in my right hand there. And then I would like to just frame for everyone that this was definitely a record-setting acquisition for the longest and slowest acquisition in startup history. And so for most of 2022, for all of 2022 and most of 2023, we were on pins and needles just sort of waiting to figure this out and toying with the idea of doing this on our own. And then finally shown here on the right is me standing with the product manager and the VP of product from CQ Medical with this display booth. And this is like 3 days or 2 or 3 days after the acquisition of the company. So it was a pretty wild ride at the very end. And I appreciate the help from LVG and from others for getting that across the finish line. So all this has given me some time to reflect on how did I get here. So I just wanted to close with a few thoughts that I have about how I got here. So one is that I think what really worked for me and what gave me the energy to push things along and the ability to get it done is that I started off with a problem that I cared about, and I just started talking to people about it and just started trying to learn more about different perspectives. I had the luxury of not having the technical skills to do this on my own, so I really from the beginning had to find collaborators and other people to work with. I feel like I found smart and trustworthy people to work with. So the BrachyFoam Advoray team, as I mentioned, Michelle and Amy were both there all along the way, and we had great consultants to work with. I also really valued not only Tim Long of Virginia Tech, but my colleagues in the Department of Radiation Oncology at UVA. It was like really tremendous to have their support along the way. And then I tried to focus on learning along the way. So I attended anything that I could get my hands on in terms of entrepreneurship and understanding how to get a product to market, offered by UVA, offered by the National Cancer Institute, and I found those resources to be really important. And then thankfully I had the support system available. So there are no telling how many people listened to me talk about this project and gave me advice over the years, not just on the Advoray side, but advisors in the local biotech community, friends and family, and probably even some people that didn't really want to know all the details, but I appreciate it nonetheless. So stepping further back, I would say, how did I get here? Having the support of Shayna and the family, including like the kids as they've gotten older, to express some interest and not give me a hard time for disappearing into the basement to get Advoray FDA materials together, has been invaluable for the process throughout this process. I'm lucky that I met Shayna with classmates in the School of Medicine, and to have a partner who understands how much work all this takes and who can relate to the clinical need is really important. Stepping further back, I would say I was lucky to be raised by two parents who really pushed my brother and I to really perform well and to expect exceptional things. And I think that was sort of the expectation at school and other domains, sports, leadership, what have you, all the way through childhood. And what I would say that's really specific to this project that I think about is like for my dad, I think my mom used to call me his shadow. So whenever he was doing a home repair or something, I would follow around and try to help. And I'm sure he gave me meaningless jobs to help him hold a screwdriver or something. But I think, I picked up some like fascination with just doing stuff and like trying to solve a problem on your own rather than like looking elsewhere for it. And so some of my childhood memories, might not seem extraordinary to some people, but like standing with my dad and like while he essentially built a tool shed, like that was a cool experience. And I think those sorts of things of like pushing forward, in addition to teaching how to interact, teaching us as kids how to interact in the world and how to work hard. I think those little things kind of like shifted me towards that realization in the Lowe's store that one morning. And then I would also like to say there's been a big focus for education for our family. I was lucky, my mom stayed home with my brother and I when we were younger, and she had to put up with us like all the time there, but really pushed education along the way. And then what's shown here is from her graduation. This is at Virginia Commonwealth University, and so she chose to go back and get an education degree, not only bachelor's and master's degree. And I was of an age where I saw like the amount of commitment that she put into it, and was able to see the rest of a really impressive career in education afterwards. And not to just so everyone despair, she did get a certificate from UVA along the way. So it's not all bad. And I think at my early age, I would just say, I found these pictures, and this was like in the bag of stuff that your parents give you when they're like ready to look at photos of their grandkids and not you anymore. But I found this in my drawer somewhere, and this is, I think, my first attempt at a science experiment sometime in elementary school. So we were living in Hilton Head, South Carolina at this time, and my parents somehow convinced the Culligan Waterman, who's shown here on the right, to go through a science fair project with me where we collected ocean water, distilled it, and this is me, analyzing my first, test tube or something of distilled water, and then analyze it for impurities at the Culligan office. I still feel bad for that guy, like he wasted a day, but it was certainly a fun experience for me, and I think my first introduction to science projects. So for future directions, so for BrachyGel, there's a pilot program launching. I left this part out, but we lost a key manufacturing partner at the very end, so that CQ Medical has had to take over some of the manufacturing steps, and they're ready to have commercial production within the next month or two, which is really exciting. They do plan to distribute throughout the U.S. in 2024. They have international distribution, so they're planning for a subsequent rollout in 2025 and beyond. For me now, this experience, going back from the early days, got me really thinking about how to make a broader impact in other ways outside of direct clinical care. So I'm still part-time clinical faculty at UVA. It's like my favorite day of the week. I wouldn't give it up for anything, but fortunately I had the opportunity to also try to make an impact in other ways. So I'm currently Chief Medical Officer of our Terra AI, which is a cancer precision medicine company, and getting to do a lot of innovation there with the first digital pathology AI test on national cancer care guidelines, which is pretty exciting. And then I'm looking forward to, in the future, contributing as much as I can to innovation on grounds. I'm very confident that at some point, once I've climbed a lot of these mountains, that I'll want to come back to UVA full time. So I think this has been a really great journey for me, and I think I'll have a lot to hopefully add to help others in the future. So just to summarize, I think this was a good and great, or great and good experience. I think hopefully it impacts patient care for cervical cancer patients, which I think is a population that hasn't had enough innovation to support their experience. I think it was great to share this experience with the UVA team along the way, as well as the team that we built at Advaray. It's taught me that I think physicians have a lot to add and to impact cancer outcomes in a variety of ways. It's not just in clinic, but I think within entrepreneurial endeavors or within industry, there's a real need to influence innovation from the clinical angle. And what I've learned, I'd say, and what is really emphasized for me for this experience is that it just really works to stay inquisitive and always try to learn. And you may find that no one really knows the answer, in which case you get the special treat of figuring out what questions need to be asked and how to answer them. And then more importantly, build a team around you and just keep moving forward. So with that, I'd like to close, and I'd like to say thank you again for the honor.
So we're going to get set up here for a few. We have time for questions. Let me turn the microphone on. But while we're doing that, something Tim said in his talk made me laugh in a flashback. Many years ago, I was a research engineer working in a consumer products company, and I did a small part on Edge shaving cream, if you know what that is. And what I was laughing was when you said this line. It was actually the TV commercial for Edge. It's not a foam, it's a gel.
So it would have been trademarked, so I couldn't have had it.
Yeah. Exactly. So with that, Whitey has the microphone there, and so if people have a question, if you just raise your hand, and we'll bring the microphone over, and we have a few minutes. So. Yes.
Tim, congratulations.
Thank you.
And that was a really good presentation. We're proud of you.
Thank you.
How did you and the team evaluate patient comfort in the study and for the FDA submission? Did you have a special tool for evaluating each patient's reaction and comforts that was objective?
So there wasn't an existing validated instrument for that, and we looked for that, but we ended up just using a simple Likert scale, essentially. So it was, for each treatment that they were awake for. So the overall treatment course was 5 treatments. For the first one, they were under general anesthesia for it, and so they would have each patient had 2 procedures where they got BrachyGel and 2 where they got gauze, and each time we asked them a series of 5 questions. What was that, 5? Okay, 5 questions about the level of discomfort they experienced for different parts of the procedure, including the insertion and removal of the packing, and they evaluated their responses on a 4-point scale. I'm looking to the PI of the trial over here, a 4-point scale for that. And so then ultimately it was analyzed, basically like a descriptive analysis of what the different trends were. So.
Other questions?
Thanks, Tim. My question is more for the actual design and device. Is it something that has to be customized for each patient? I'm not a physician. I don't know if each person has particular intricacies in the anatomy that you have to be mindful of when you're designing it.
Thanks, Courtney. So the catheter itself is like a one-size catheter, and some details about that catheter is that it's designed, there are essentially 2 channels. So one is a larger channel where the hydrogel precursors get injected into it. It can handle a fair amount of volume. And then the other channel has a saline port, so you can actually remove a plastic filament at the end of the case, and there's a channel in the solidified gel, and you can just insert saline. The bag itself is designed to be oversized. So it's an 80 milliliter reaction bag with very thin walls. And we inject in the hydrogel material, which is about 50, 60 milliliters. And the idea is by being oversized, that gel can expand, and it's the expansion itself rather than the physical properties of the therapy bag that determines the ultimate orientation of the gel. So that was intentional to sort of replicate expanding within the cavity. And so the way that it looks different for each patient is that the clinician may only based on their examination may not inject the entire contents of the syringes into the bag, or may choose to use 1 versus 2 bags, and then the expansion, the way that the gel expands, is customized to that patient too.
Do you envision BrachyGel being useful for other types of cancers outside of cervical cancer?
That's a great question, and that was in the early days something we spent a lot of time thinking about for other options. I do think so. The most obvious thing to me is, I will admit, sort of a niche brachytherapy area for GYN cancers, but we had a borderline score that didn't quite get funded for another STTR grant for use for essentially adjuvant therapy for endometrial cancer. So that would be a different applicator system. It would call for a slightly different delivery system to make that work. And then I think this sort of technology might be useful in an adapted way for a surface application, like for external beam radiation therapy for skin cancer. But I couldn't think of much else that we could do with this sort of delivery system. I think hydrogels certainly have a great role in our field. I'd say the most famous and widely used application of hydrogels that have a pretty similar chemistry, or at least material composition, is the Boston Scientific's product called SpaceOAR, which is a permanently injected device that dissolves over time that creates a space between the prostate and rectum. So I felt like, I think in terms of like commercial opportunities, this represents probably the second largest impact behind the prostate cancer tool that I think could be made. And then there's probably a couple other applications. I don't know if I have the energy to carry those forward. Maybe someone else from the UVA team will do those. So.
Probably have time for one more question if anyone has something burning that they haven't asked yet. Mr. Decker.
Thank you, Dr. Showalter. So during this journey, obviously you had a great idea, which really meant thousands of ideas in this area. I was just curious, did you come up, during this journey, did you have other ideas in other areas that you said, oh no, there's no way I'm going to touch another idea and just run from it? Did these come up? What happens if you have another idea in another area? Just curious what you're going to do with it.
Well, just as an example, sometimes you have an idea, it seems great, and then you fall out of love with the idea. You realize there's some fundamental flaw. And I'd say those two examples that I just provided in terms of the vaginal brachytherapy application and the surface bolus application, I think are ideas that could make some clinical impact, but would take a lot of work to get through and may not be large problems to take on. I'm interested, after doing this, I would say what was fun about it, and I think what gave me the chance to learn about it, was about a small area that I knew a lot about, and I knew the patient narrative and how it would improve care, and I was really expert at the time, at least in that procedure, and I feel like I could wrap my head around it. I think I'm interested now in taking on larger problems. So as I mentioned, I transitioned into a position now, which is most of my professional effort for a company that's going across solid tumors. And I'm always thinking about new ideas. I think that one thing that I've learned is that everything's hard and everything takes a long time, and you might as well really focus on big ideas. So I think my plan is to take what I learned from this for maybe a smaller, more circumscribed area, and use those lessons from the regulatory and manufacturing, all those steps, to really make an impact on something that touches a larger number of patients each year. So,
Thanks very much, Tim.
Thanks.
Josh Malden, would you come up here, just give me a hand? Josh is our Director of Licensing. So Tim, on behalf of the Licensing and Venture Group, on behalf of University of Virginia, I wanted to congratulate you on being the Edlick Henderson 2023 Innovator of the Year. Congratulations. Thank you. We have one more thing? Yep. Yes. Oh, wow. It's attracted a lot of attention walking across grounds today. So the prize does come with a check for $10,000, so congratulations.
Thank you so much.
You put it in your purse?
Hey, Rich.
Yes, thank you. So here we have our plaque of our prior Henderson Innovator of the Year winners. So we're going to ask Tim to add his name to the plaque. So Tim, here you go. Please, after the talk, we were joking that this man's a doctor. He's got to have good hands for this without shaking. So, all right. So thanks very much. That concludes the formal part of our program. Downstairs, we have a reception where we have some drinks, some wine, and some food, and I hope you'll join us and Dr. Showalter and his family in celebrating the event. So thanks everyone for coming.